Allow me to speak in two languages for today. We have Arabic speakers here also. بداية السلام عليكم. حبيت أرحب بكم في هاي المحاضرة الليلة. أنا أول شيء أعرف عن نفسي. أنا اسمي محمد الصايغ، باحث علمي إماراتي في مجال علوم الأحياء الجزئية أو اللي يسمونها في الإمارات جينات. واشتغل باحث ما بعد الدكتوراه في جامعة نيويورك، في أبو ظبي وفي نيويورك. اليوم حبيت إني أول شيء أشكركم على تواجدكم في هاي المحاضرة اللي تعتبر من أحد محاضرات جامعة نيويورك في أبو ظبي، واللي تأثر على دور المجتمع الإماراتي. واليوم محاضرتنا اليوم عبارة عن جزء مهم في مجتمعنا اللي هو مرض التوحد. ومرض التوحد مثل ما أنتم عارفين يعتبر من الأمراض اللي قاعدة تنتشر في دولة الإمارات من الناحية الوراثية. واليوم اللي بيتحدث أو الضيف اللي بيساعدنا في فهم هالموضوع من ناحية وراثية راح تقدمه الدكتورة كريس غونزاليز، اللي هي تعتبر مدرس مشرف بين جامعة نيويورك في نيويورك وفي أبو ظبي. Okay, for the English part. Okay, allow me to introduce myself to you. My name is Mohammed Al-Saigh. I am an Emirati molecular and cell biologist working as a postdoctoral researcher in NYU Abu Dhabi and also in NYU. Today, I would like to welcome you for coming for this talk, which reflects its importance on our society. So, the talk today it has a really close interlink with our society, in that the society here suffers from autism at a genetic level. And our speaker for today, who's an expert in this field, was very generous for him to come to the UAE and share his thoughts with us, who will be presented by Dr. Chris Gonzalez, who's an affiliate faculty at NYU in New York and in Abu Dhabi at the biology department. So without further ado, allow me to welcome Chris Gonzalez to the podium.
Thank you very much. It's my great pleasure and honor this evening to welcome Dr. Eichler. Evan is a Howard Hughes Medical Institute researcher and a professor at the University of Washington in the Department of Genome Sciences. He is a world-renowned leader in the field of understanding genomic variations, so ways in which chromosomes become rearranged. And I first met Evan at a meeting in Berlin, where he was invited by another colleague, and I was totally blown away by his work that I went off and studied up and read all of his papers. And it's really amazing what he's been able to do in this field. So just to give you a little bit of background, he received his undergraduate education in Canada, in Saskatchewan. He's Canadian originally. He then received a PhD at the Baylor College of Medicine, where he studied a gene that's associated with a mental retardation. It's called Fragile X mental retardation gene, FMR1. And specifically what he was looking at was how disease phenotypes are associated with the number of repeats of a small 3-nucleotide repeat within this gene. And this is actually a very common type of variation that's associated with diseases and other genes as well. And you may have heard of another Huntington syndrome. And this is, for example, analogous to saying, instead of a sentence where I just say the something or other, it's a defect in which I would stutter or say the something or other. So for lay people who aren't geneticists. And what he's been able to do is really to pioneer methods to identify and study rearrangements and duplications in genomes. He's pioneered methods to understand how to infer the history of repeats that we can see in genomes, and also to try to understand their relevance to human disease. And for example, one of the things that he can understand is when we see particular rearrangements in the human genomes, we can see if those were shared by our last common ancestors or if they're unique to humans, and also whether they're unique to specific individuals. And it turns out that most of us have some private genome rearrangements, that now that we have so much ability to obtain sequence data, we're starting to learn more and more and more about these things. But I think in general, our ability to collect data really is far exceeding our ability to analyze and interpret those data. And I just want to give you one personal vignette about Evan and an experience that I had, if you don't mind, which is that I had a colleague at NYU who was expecting a baby with his wife, and they did some genetic testing, and they identified, it was discovered that his fetus contained 2 small extrachromosomal fragments which were derived from a couple of chromosomes, but they were very, very short. And they tried to speak to some genetic experts in the world. They got advice from their physicians, and some of their doctors were advocating that they might want to consider aborting their fetus because they didn't know if these genetic abnormalities were going to result in some very serious, debilitating developmental defects in their child. So he contacted me at the suggestion of a friend, and I told him that I really can't help you with this, but I know the world's leading expert in this area, Dr. Eichler, and with his permission, I forwarded all the information to Dr. Eichler, who, of course, said, I can't give you medical advice, but he passed the data within an hour. I had received an answer from him. He had given the data to his postdoc. They had looked through their very extensive database of genomic rearrangements, defects, abnormalities, and he said, well, we have some data that suggests that very long duplications in the regions of the chromosomes that you're talking about have some deleterious effects, but we don't have any data on these very short rearrangements, which are probably personal rearrangements in this child. So my friend agonized for a long time over what to do about the possibility of raising a very severely disabled child. In the end, he and his wife decided to go ahead with their pregnancy. And they were very pleased to announce, and I also forwarded his announcement to Evan, that they seem to have given birth to a perfectly healthy, lovely young baby recently. And they don't know she'll have developmental defects down the line, but so far, so good. 10 fingers, 10 toes, no obvious problems. And I just want to show you a picture of this beautiful baby that he sent me. And so in addition to being a really spectacular scientist and researcher, he's also an extremely kind and humane individual. And so please, Evan, we would love to hear your talk.
Well, thank you, Chris, for those very kind words. And I actually feel like I have nothing left to say. I mean, that's essentially what it's about, right? It's about the children. And everything I'm going to tell you today, although I do a lot of evolutionary work, as she mentioned, will be focused on essentially our work as it relates to autism and our hope that we'll be able to improve the lives of autistic children in the future. So what is autism? I think many of us have our own idea of what it is. It's broadly defined as a neurodevelopmental disorder, and it's recognized, at least it was recognized, as showing impairments in really 3 areas. Impairments in social interactions, repetitive behaviors, and usually fairly severe speech delay. It falls under a category more broadly defined as developmental delay, which afflicts, in terms of the moderate to severe, about 2 to 3% of all births. The most recent data from the CDC suggests that as many as 1 out of 6 children are afflicted with some form of developmental disability, so inability to make certain milestones. So in terms of the autism definition that people look at, the estimates from the CDC are now 1 out of 88 births. I think it's important to note that there's very little evidence that there's an ethnic predilection, so it doesn't appear that there are populations that are particularly enriched by multiple factors. It's a human-wide condition of autism. It affects every population, every tribe, every group of individuals on the planet. One of the interesting aspects that's been shown over and over again, and is more broadly applied to a whole range of neurobehavioral and neuropsychiatric disease, is the fact that there's a strong gender bias. So developmental delay, more broadly defined, is about 1.3 males for every female that's affected. Autism is estimated about 4 males for every female. And higher functioning autism, which used to be described as Asperger's syndrome. So these are kids that clearly have a high IQ, but they have a lot of social impairments as well as repetitive behavior and speech issues. 6 males for every female in terms of the disease. We don't have a great explanation for this difference in terms of the gender, but I'm going to show you some data which I think sheds insight on that today. In addition to the economic issues and the personal issues, which I think are tantamount, there's also a significant financial burden. The estimate in the US right now to essentially raise a child with autism for its entire life is about $3 million for the individual. So it's a significant issue for all society. So this is just some of the features of autism that have been associated or described. And you can see here, I think most of us know or have in our own family an individual with autism. And the important point here is that there are certain aspects of this that actually are pretty characteristic of the disease, such as inappropriate laughing and giggling, avoidance of eye contact, echoing words and phrases over and over again, difficulty in expressing needs, using hand gestures as opposed to language, and difficulties with interacting with others. Not every one of these aspects is seen in a child with autism. Usually it's a subset of these. And it's more complicated than just these types of behaviors. Often there's comorbidities with diseases like epilepsy, within families schizophrenia, other types of diseases that are not defined as autism often are associated with families with these diseases. What I'm going to focus today on is the genetics of autism. I'm going to tell you about some of the observations that we and others have made over the last, I guess, 7 or 8 years. First, to give you some background about the genetics of it, why we believe it's so important. It's been recognized for at least 30 years that there's a strong genetic component. It's now estimated that about 50% or more of the risk is actually genetically controlled or based. And it comes largely from identical twin studies. So if you look at identical twins versus individuals who are brothers and sisters, what you find is that somewhere on the order of 75 to 95% of identical twins are concordant. That means if one twin has it, the other twin will have it. For brothers and sisters that are from different eggs and sperm, the number is about 5 to 20% in terms of essentially concordance, which, if you remember, is significantly higher than 1 out of 100 births, right? So the risk, if one individual in the family has autism, that another child will be born with it is about 5 to 10-fold higher. So these have all suggested that there's a very strong genetic component. The really disappointing thing in the 1990s is that huge amounts of money were spent in the US to try to identify common mutations that might be associated with the disease, and pretty much came up empty-handed. So almost very little information, despite spending many hundreds of millions of dollars in terms of trying to identify genetic causes. We knew that within families, that about 8 to 10% of autism could be explained by transmission within families, so people could do classic genetic-based approaches and track down the genes. But those types of families in which there's a strong familial predilection going through multiple generations are relatively rare. So the question that's faced our community is, where's the rest? Where's the genetic cause? If there's such a strong signal coming from identical twins, where are the actual genes and the mutations? A little bit about the pedigrees of autism families. So I'm going to use this notation a lot today just to kind of indicate. So you're looking at a 3-generation family. The individuals affected with autism are highlighted in black. Males are squares, females are essentially circles. And this is a nice example of a familial autism case. So here you have an uncle, a grandson, and a granddaughter, all afflicted with autism in that particular family. So this is a minority. This is about 15% of all autism cases have familial and this clear of a signal. More often we see things like this. We call this out-of-the-blue autism or sporadic, where there's essentially no signature of family history, and all of a sudden we have a child in his 3rd generation, a grandson, born with autism. Turns out that the story is a little bit more complicated. More often than not, pedigrees look like this. So if you really study them in detail, while this is a strict definition of autism, meeting the criteria that I mentioned regarding social interaction, repetitive behavior, and impaired language, often we see pedigrees that look more like this, where we actually have one child, only one child, a grandson here with autism, but we have a mother who may have some kind of attention deficit, we may have a grandfather who had schizophrenia or seizures, and we might have a cousin who essentially has intellectual disability. And so this comorbidity is a real complication to studying this disease because geneticists like to find genes and clinicians like to put things in bins. And so we often, unfortunately, our genes don't necessarily read the same manuals that the clinicians are reading. And so we have to account for families looking like this and like this more than this in terms of trying to identify the genes. So here's what I want to try to accomplish tonight. I want to give you some insights in terms of the role of genetic variation in terms of autism, how we started looking for really unstable regions of the genome, what I will call genome structural variation, and how we use that to begin to understand developmental delay more broadly, some of the genetic causes of it, and how that led into research with respect to autism. I want to share with you some of the new methods and technologies that have been developed now to systematically characterize the exomes and genomes of families with autism, and I'll explain some of these terms as we go. And then I want to share with you some of the real challenges that we're facing right now trying to prove specific genes so that we can make a difference in the lives of families and say for sure that a child is going to develop autism or for sure a child will not develop autism. That's probably the hardest part in terms of the work that we do. And then I'll share with you some of the future directions and implications of the research from our group and many others from around the world in the last 10 years. And hopefully convey to you why I'm so excited about the future of this field and the importance that we can possibly make with respect to this disease. So as Chris told you about, I'm going to talk about genome structural variation. So I'll start with a definition of what it is. So this is all examples of genome structural variation. What you're looking at here are chromosomes, one from, let's say, your mother, one from your father. And what's different, this is one particular chromosome of the 23 pairs that exist in our genome. This individual has less material for a specific region of that chromosome. We'll call that a deletion. This individual, for example, on this particular chromosome, has an extra copy of everything that sits here. We'll call that a duplication. And just to be comprehensive, and I won't talk anything more about this, we can also have within our genomes changes in the order. So where the genes become flipped, and we'll call that an inversion. So this is kind of the definitions of genome structure variation. All of this deletion and duplication is called copy number variation. And so I'll use that word a lot today, copy number variation or shorthand CNV, and what I'm referring to are both deletions and duplications, typically big events. So most events I'll describe today are hundreds of thousands of base pairs in size, so not small mutations, at least with respect to this type of mutation. All right, so where to begin? So as I finished up in grad school, I had the great opportunity to actually participate in the Human Genome Project. And I think I was forever changed as a result of that experience. So many of you may or may not know, the first human genome, which was actually a conglomerate of many genomes, was sequenced about 2001. A higher quality version of that sequence came together in 2004. It was an international effort with thousands of individuals involved in it. And I had the opportunity to actually look at the genome and study it for various attributes kind of for the first time, at least for my own interests. So the goal of the Human Genome Project, and it was a very expensive project. Some estimate it was almost upwards of $1 billion to do the first human genome. The goal was essentially to find all of the base pairs of sequence that actually constitute the chromosomes. So basically to decode the genetic code and to identify all 20,000 genes and where they sit within the actual chromosomes. So that was the mission. The sad news is that we're not still done after 14 years. There's still work to be done, but it's 99% done in terms of these two things. My particular role in that project was actually to look for regions of the genome that could be potentially unstable based on the organization of the regions of the genome. So what I was looking for were hotspots of potential genetic instability such that every time those chromosomes were transmitted, there would be a risk of mutation as a result of the genetic instability. And so that required looking at things comparatively, as Chris mentioned, but also looking for essentially features of the genome. So this is a complicated slide, but it's one that we produced in 2002, which is essentially guided our research afterwards. And so what I'm showing you here is a schematic representation of all of your chromosomes. And the blue lines represent very large repeat sequences that exist at multiple locations on specific chromosomes. And so you can see them scattered across different chromosomes. So each time you see one joining blue line, that means there's a big repeat that sits at one location and another location on that chromosome. Everything that you see here in gold represent areas of the genome that we believed or we predicted would be genetically unstable. And that was because they had really the right type of repeats and the right structure of repeats in terms of these blue lines around the given region of interest. Everything that you see with a letter behind it, everything from A, I think, to about X, represented regions in 2002 that we already knew, when mutated, deleted, or duplicated, were associated with developmental delay. So we had already at that time, we knew about 20 different disorders that were resulting from genetic instability of these hotspot regions. And so we identified another 100 such locations in the genome, representing about 10% of all of our genetic code. And we predicted that these regions would basically be genetically unstable and may explain essentially a significant fraction of developmental delay. So that was the hypothesis that we had in 2002. So just to be clear what the hypothesis was, we knew back in 2000 that in fact most children that were born with developmental delay, and more specifically autism spectrum disorder, there was no genetic diagnosis. So about 80%, some estimates up to 85% of cases that would come into a clinic, no one, after running all the DNA tests that were available at that time, could come up with a diagnosis. So some of them you're familiar with, like Down syndrome, Fragile X was mentioned, other types of things. But in most kids and most families, a child was born, didn't pass developmental milestones, would go to be tested, and 80% of the time the family would get a result and said, we don't understand why your child is the way the child is. And so we knew that essentially clinical labs were full of cases where there was no genetic determination of what the cause was. And so our hypothesis was, could we go back to these regions, these hotspot regions that we got from the human genome sequence, and could we test those patients specifically for deletions or duplications, i.e. copy number variation, over those specific hotspot sites? So our model, which was naive, was that the parents would be truly normal, and they'd have just 2 normal copies of each chromosome, which all of us hopefully have. But in the child, that there would be something that would happen in the production of either the egg or the sperm that would result in a big swath of sequence being deleted or duplicated. So we were looking for things like this, where the child looked like this and the parents looked like this with respect to their chromosomes. We were lucky that essentially technology had already come on board for studying cancer, which was called microarrays, which allowed us to really do this in a very systematic way. So what I'm showing you here is essentially a portion of a glass slide, and this is the dimensions, 12 millimeters more or less by 15 or 16 millimeters, where every dot here represents a DNA molecule that's actually been fixed and synthesized and put at that specific spot. And so what we could do is we could build these microarrays for the exact sequences that we predicted to be genetically unstable, those genomic hotspots. And then what we could do is we could actually label the DNA of a patient with one color. So I'm showing you here it labeled with green. And label another individual, let's say a parent or a normal individual without disease, with a different color. And if you label these DNA, you color them, and you throw them on top of this chip, the DNA knows where to go and actually will hybridize to the location where it has essentially the highest identity. And what you can do is you can actually measure the relative intensity of both the green and the red. And if they're equal, there's no difference between the normal and the patient. If they are different, so the green intensity is higher or the red intensity is higher, that indicates you actually have a deletion or a duplication. And so the power of this technique, it was borrowed from the cancer field, was essentially applied to children with developmental delay, and we could put specifically on this chip the sequences corresponding to the hotspot regions and ask the question, does a child have a deletion or a duplication for that specific region? So we knew the address of every one of these in the human genome, and we could screen thousands and thousands of sites and thousands of patients based on doing these experiments one at a time for every patient. So that's the work that we did. I'm not going to bore you with any more details other than that to just give you a sense that this was an example of the new technology that arose from the Human Genome Project, but really to share with you some results. So we gathered, when we first started this in 2006, we had our first 300 patients where everybody had said, we don't know what the problem is with these children. They've been tested. There's no explanation for why the kids have intellectual disability. And in the first 300 patients that we tested, we found 4 that had a particular region of chromosome 17, only about 500,000 base pairs, which might sound like a lot, but when you think about the genome being 3 billion base pairs, it's not very much. They had this particular piece of DNA deleted. Their mom and dads were totally normal. The child had 1 normal chromosome and 1 chromosome where those 500,000 base pairs were missing. We identified 4 children out of 290 that actually had this. And when we brought those kids back in to the clinic to study them, what we found was that the kids had very similar features. So these are all kids with developmental delay. You can see the really characteristic facial features, kind of this protruding tongue. If you look at the nose, the clinicians recognize the nose right away. It's very bulbous. It has almost like a little bulb aspect to it. And you can see, maybe you can't see, but these kids are extremely happy. So when they come in, people have always commented on it. There's now hundreds of families with this particular deletion. The children are always happier than everybody else around them. So they're some of the happiest children that the clinicians actually ever meet, they say. And all of these were essentially of the same type. And so the power of this, if you think about it, so we found it, there were 2 other labs that found it at the same time. The power of this is that we had now found a new region of the genome, when deleted, which was associated now with a very specific type of developmental delay. So if we have this big umbrella of developmental delay, we just identified a different type or subtype based on this deletion in these kids. So that was one example. A year after, we found another example. This one, different region of the genome. Again, one of the genetically unstable regions that we predicted. These kids all had a piece of DNA, now about 3 million base pairs on chromosome 15, that was missing. Parents were normal. Parents had both copies of that sequence present. And these children had a different phenotype. All of these kids were classified as autistic. They met criteria for autism spectrum disorder, but they also had facial features. You can see this long forehead. You can see the almond-shaped eyes of these children. They all had the same lesion, relatively rare. We think it's about 0.1% of autism, but essentially now there's about 24 families that have been described worldwide with the same type of event. Important point here is that we screened thousands and thousands. Now we screen close to 30,000 normals, people in this room, more or less, and we never see these events in the normal population. So this is almost like 100%, right? You have this deletion, you're going to have a child. One thing you can't see is these kids are in the 25th centile in growth. So pretty much every aspect is recognized early on for these particular kids because they're actually very small in terms of their growth trajectory. This last example I show you is one that actually we did not consider to be disease-causing. This is one where the clinician said there's no obvious facial features here. All of these kids had a deletion in a different region of chromosome 15, but there was no obvious facial features that you could link them. One interesting aspect of all of these individuals that we identified was they actually, in addition to having borderline intellectual disability, they also had relatively severe seizures, and often. So we initially didn't pay much attention to this, and I tell you why we didn't pay much attention to this one. Because our initial index case shown here actually came from a family where this deletion was transmitted from the mother. And remember that model that we had? We're looking for new mutations, so sporadic occurrences. In this particular case, it didn't fit the model because it was transmitted from a mother. We knew when we screened lots and lots of normals that we didn't see the deletion, but the clinicians had told us that the mother was normal. So we kept on screening. We screened another couple thousand patients, found 2 additional cases, and in both of these cases, they were sporadic de novo mutations. So i.e. the lightning bolt means they're not mom and dad, they're a new mutation in the child. So what we did is we said, well, can we go back to this family and have the clinicians reassess the mother, the father, and anyone else in that family? And this was the result. So it turns out this is the mom of this individual. This is a half-sister of this individual. And it turned out that when we went to the pedigree, that essentially this individual and this individual were both affected. So these are the 2 sisters that I'm showing here. The mother was borderline. She had a job. She never graduated from high school, but she actually had some learning disabilities, but not anywhere close to severe as the two daughters. She had been married twice, and from her second husband, she had a normal daughter which did not receive this particular deletion. And so taken together, this is now one of the most common causes of epilepsy and intellectual disability in the human population. But it started with essentially the idea that you could be both now de novo, i.e. sporadic in cases, or you could be inherited. And so this is a really important point. Just because it's not, or it is found in a parent, does not necessarily mean it's not relevant with respect to disease. This is even a little bit more confusing. After we published this initial study reporting this deletion on chromosome 15 associated with intellectual disability, a study came out literally 6 months later reporting this deletion as one of the most significant risk factors of schizophrenia. A few months later, Tony Monaco published a paper where he showed this particular deletion segregating with autism in a family. And then we worked with a group out of Germany to show that about 1% of individuals with epilepsy have this deletion, not necessarily intellectual disability, but have this deletion compared to 3,339 ethnically matched German controls who did not have this particular deletion. So this is interesting and also a bit confusing, because here we have the same mutation, but manifesting very differently in terms of diseases. And our current hypothesis is that, in fact, it depends where this mutation finds itself. It depends on what genetic background this mutation is found to dictate in terms of the outcome with respect to disease. So this is a little bit of a technical slide, but I thought I would share it with you. So kind of putting it all together at the end, going back to those hotspot regions that we identified in 2002, it's now about half of them, there's about 55 of them, that are now associated with recurrent microdeletions, largely microdeletions, but also duplications and developmental delay. And a subset of those are associated with autism spectrum disorder. And it turns out that this model of copy number change is actually much more important than we have first thought in terms of actual disease. So what I'm showing you here are the results of a study published a few years ago where we looked at roughly about 10,000 normal individuals and compared it to 50,000 cases of children that came into the clinic with unexplained developmental delay. And what these curves indicate is the proportion of the population, shown here in the percentage, that carry a CNV, either a deletion or a duplication, of a given size or greater. So the way to read this is that if you have a deletion that's 500,000 base pairs in size or greater, 25% of kids that come into a clinic now for testing will come back with a positive result, compared to the people in this room where it's about 10%. So 10% of you carry a large deletions or a duplication, but if you went to a clinic with pediatric cases of unexplained developmental delay, it's going to be 25%, suggesting that as much as 14% of the, quote, genetic cause, unquote, of developmental delay is the result of these large CNVs. All right, so is that clear to everyone in terms of what we're looking at here? So the idea is that from a kind of a global perspective, large-scale deletions and duplications, and largely deletions, are contributing significantly to developmental delay and autism. The numbers are closer to about 10% for a strict definition of autism. For developmental delay, it's higher, it's like 14%. There's one little twist on this, which I'll share with you. In cases where we've been able to go back and look at the inheritance of these CNVs. So remember, there's two models. New mutations, which are occurring either in the production of the sperm or the egg, or we could have inheritance, like that chromosome 15 region that's being transmitted. What we find is when we look at families with developmental delay is that females tend to have more CNVs than males. It's a significant finding. We find that disease-causing events, at least the things that we consider risk factors for disease, are more likely to be transmitted from mothers compared to fathers. So 60% of the time coming from mothers, 40% of the time coming from fathers. And the data suggest is that females are actually protected and have a higher tolerance to these types of mutations than males. And I'm happy to discuss why that might be the case. We don't know for sure why, but the data seems to be now fairly convincing that females are more likely to be carriers and unaffected when compared to males, which may help to explain some of that gender bias that we saw in the very beginning. So what did we learn from CNVs? Well, large deletions and duplications are very important in terms of at least a subset of the genetic basis of autism and developmental delay. But more importantly, it suggests gene imbalance. So not having 2 copies of a given gene, or having maybe 3 copies where you should have had 2, is in fact, for some genes, a big deal, and actually can result in contributing to disease. These events can be inherited or new mutations, i.e. sporadic, and we think that's happening when eggs and sperm are being produced. So not really transmitted in the sense of essentially moms to offspring, but more often these are new mutations that are cropping up every time that germ cells are being produced. They account for about 14% of developmental delay, 7 to 8% for autism spectrum. Mothers are more likely to carry and to transmit these. So CNVs have been fantastic for helping us to identify a model for thinking about the genetic basis of autism, how we have to have new mutations, very young mutations in terms of contributing. But they absolutely suck from the perspective that they don't really identify the genes. And the problem with big CNVs is that they're big. And so they typically involve multiple genes. So shown here are the genes with a little, in the stick diagram here. And so when you have a deletion that's recurrently deleting this segment, there may be, let's say, a dozen genes in there. So you don't know which specific gene is responsible. Or maybe it's more than one gene that's responsible. Or the same thing for duplications. So this takes us up to about 2007 with respect to where we were in terms of thinking 2008. And something very important was happening in terms of genetics around this time. Some of you may be familiar with this, but a new technology in terms of how to sequence genomes was coming on board. It was called next-generation sequencing. And the technology was significantly different than what we had done in the past. But the important point, the really important point, is that we could sequence genomes essentially orders of magnitude faster than we could before. And perhaps most importantly for research studies, we could do it for a lot cheaper. So shown here is the cost on an exponential scale for sequencing a human genome from 2001 to 2014. This big point right here is when the new generation, next generation sequencing technology started to come online. So in about a matter of 3 years, we went from around $10 million to sequence another human genome down to about 10,000. And this number is a little bit optimistic. It's not quite there yet. It's closer to about $2,000, even with the newest technology that has come out. But people always want to actually draw the line a little bit farther down, I think, than it really is. But the important point here is that the cost of sequencing went down significantly. So if we go back to that question about sequencing genomes, even when you're sequencing for $1,000 or $2,000 a genome, it's still pretty pricey when you're talking about an individual family. So you want to sequence a mom and a dad and a child, that still costs you $6,000, and you add additional things in the clinical setting on top of that, you're quickly talking about $12,000, $15,000 for a given family to actually sequence their genomes, with a chance maybe of finding a mutation. So one more thing happened around 2007, 2008, and it was a shortcut. So instead of actually sequencing all of your genetic information, what people decided to do, and I actually criticized this at the beginning, but now I actually embrace it. Now I think it's actually fantastic, was that instead of sequencing everything, just sequence the portions that will actually encode the proteins. So in your genome of 3 billion base pairs, only about 1 to 1.5% of the genetic code actually is recognized as producing transcripts that will actually produce proteins, which are really thought by most people to be the functional portion that makes the cells in the developing individual. And so they came up with an approach to actually sequence, instead of the entire genome, just sequence the protein coding portions, which we're going to call here exons, and skip all the rest. And so if you think of it like a big apple, we're just sequencing a slice. And so this could be done for a few $100s as opposed to a few $1,000s, making it much more tractable to really sequence families just for the protein coding portions. So the hypothesis coming from the CNV work, the deletions and duplications, could we go back to those families where we haven't actually found a mutation? in a big deletion, which is the majority, right? So still 70% of the families we don't have an explanation. Can we go back to them and now sequence just the protein coding portions, or the exome? Can we sequence a mom and a dad and the affected individual and look for new mutations that would have essentially the same result, essentially killing the gene? But this time we're looking for very small mutations, things that would actually disrupt the gene, have the same consequence of eliminating it, but just essentially like a sliver in terms of the size compared to these big deletions and duplication events. So that was the hypothesis, and the goal was to get gene specificity. So while the CNVs told us that we had regions of the genome with multiple genes, this in principle would actually get us to the actual genes. If we understood the genes, maybe it would tell us something about the biology of autism. Maybe it would tell us something about how we could develop therapies going forward with respect to these specific genes, and identify another subset of the genetic cause. So 2012 was a banner year, I have to say, for sequencing autism families. There were 6 studies that involved intellectual disability and autism, and there were trio studies, and about half of them involved sequencing a mom, a dad, a child with autism, as well as an unaffected sibling without autism. And we call those quads. So there was about half of them were doing just trios, so mom, dad, and child, which is affected, and half were essentially looking at, in addition to that structure, an unaffected individual. So about 1,000. Ours was one study of about 210 families, and the others, everybody contributed, and they were published. And the results for autism were exciting and also scary. So the good news that came from sequencing, my colleagues would kill me for actually reducing all of this to one slide and essentially not going into detail on these papers, but I think the important point is here. The really good news that everybody was so relieved is that essentially when we sequenced this individual, compared it to his mom and dad, and sequenced this individual and compared it to mom and dad, and looked only for the new mutations, the number of mutations that were between this guy and this guy were about the same, but the mutations were very, very different. We saw in the affected individuals with autism a 2 to 3-fold increase in the number of new mutations that actually kill genes. So just like the CNVs that break and remove a copy, we were finding a 2 to 3-fold excess of gene-killing mutations in the kid with autism compared to the kid in the family without autism. So that was really exciting. And that signal's not going away. It's been done now at the level of 5,000 families, and the signal is strong as it ever can be, suggesting that there's going to be strong genetic signal in new mutations in specific genes. This was the bad news. When we put all of our data together, because we were hoping for maybe 10, 20 strong autism genes, we almost never saw the same gene hit twice. So we found 18 genes when we combined all this data together that were seen where there was 2 independent new mutations seen in different families. And this is actually significant, so much more is unlikely to have occurred by chance. But the bad news is we could estimate how many genes were going to be responsible for autism based on this recurrence rate. And the estimate is between 500 to 1,000 genes responsible for autism, at least in this model of just simple new mutations that take out a gene or a conserved function in that protein. So this is sobering because it means that a clinician or a doctor that's been seeing autism patients for 50 years has probably never seen the same genetic cause of autism twice. So it's always been a different gene in a different family. And it means that all the sample collections that we had at this point, 1,000 here, 1,000 there, are going to be totally underpowered to really prove the genetic, that any of these genes are actually responsible. So even though we have a strong signal of a 2-fold excess, half of those genes are irrelevant with respect to autism, and half of them are important with respect to autism, right? In our own study, there was something else that came up. So this is my chance now to say something negative about the dads. So mothers, if you thought I was being unfair to you guys, now it's the dad's turn. So when we looked at the new mutations, what we found, and this is in our own study now of 209, we had the ability to determine where the new mutations were coming from in about 20% of the new mutations, because there was another genetic marker close by that we could assign essentially the new mutation to either the mom or the dad. And what we found is that 80% of all new mutations are coming from dads. So dads, you are responsible for 80% of new mutations in the human genome. In a study that followed shortly thereafter, it's been shown at the genome-wide level that every individual on average has about 40 new mutations that aren't in their mother or father, 40 to 50, and 80% of those are coming from the dad. Interestingly, if you consider the age of the father at the time of conception, there is in fact a significant, it's modest, but it's a significant correlation with advancing paternal age. In other words, so this is the age of the father at the time of conception. This is the number of individuals that had 3 mutations in the protein coding. These are the number of individuals that had 2. These are the number of children that had 1. These are the number that had 0. And you can see there's this trend line going up. It's significant. In other words, older fathers will tend to have children with more new mutations than younger fathers. And this actually fits pretty nice with the epidemiological data, which suggests that a risk of a 45-year-old father having a child with autism is about twice of what it is for a 20-year-old father. There's other reasons why 20-year-olds shouldn't be fathers, but I'm not going to go into that. And it's actually, when you think about it, if the risk is 1 in 100, most fathers that are 45 are not going to have an autistic child. Don't walk away with that message. It's still, the risk is only 1 out of 50 of your offspring, and most of us don't have 50 kids, so it's not really such an issue. The second issue, and I should point out that this has actually been predicted by theory for many years. It's just that we have now the observation. It's not specific to autism. It's just a general property of advancing paternal age, and that's because the sperm mother cells actually go through more divisions as the father gets older. And every time you go through a division, you have an increased risk of actually having a new mutation in the sperm cell. The other point, which we thought was very exciting, and actually I didn't believe, so I have to basically give credit where credit is due to my postdoc. So what he did is he looked at all of his mutations and said, well, I don't have any recurrent hits, or very few. He had a gene called CHD8. We had 2 loss of function mutations. He said, if I take all of my genes that I see in my autistic kids, and I look to see if they produce proteins that interact with one another, is there any relationship about the genes that are actually producing proteins? So at the protein level, is there any relationship among these mutations that we're finding, the genes that are being hit in the autistic kids? And what Brian found out is that when he did this, about 40% of his new mutations actually produced proteins that were part of one highly interconnected network. So what does this mean? What you're looking at here are, from our 209 families, the new mutations that we're seeing. And these are the proteins that are being produced. And every time you see a line joining something, that means this gene is producing a protein that interacts with this gene that's producing a protein. So at the protein level, these genes are interacting. And that's because most genes produce proteins that don't live in isolation. They work with other proteins to build cells, to control development, and so on. Everything indicated in color is a gene-killing mutation. So we would think that there's half of the copy of that protein produced. And everything seen in black is a very conserved area that hasn't changed over evolutionary time that we now have seen mutated in a child with autism. So was this significant? Well, the network was more interconnected. I joke this looks like a kind of a ball of yarn, right, in terms of all the connections. It's more interconnected than you'd expect by chance. It's already, when we did these tests, we know it's enriched for genes that we already know are involved in autism. And when we look at the new mutations in unaffected siblings, we don't see this network. So that was all a good sign that this network might be important for thinking about the biology of autism, how it emerges. All right, so back to this problem. 500 to 1,000 genes. So every family has their own, that we would see in a clinic, would have their own genetic cause and principle of autism. So I was lucky enough to be in a department where new technology was being developed to essentially exploit some of the new next generation sequencing, and in fact further reduce the ability to actually target just specific genes and to do it in very high throughput, high throughput and very cost effective way. So I'm not going to go into detail on this, but this is for the aficionados that might be interested in this. The technology is called molecular inversion probes, but the bottom line, what it allows us to do is target 40 to 50 genes just for the protein coding portions, and to actually do this in a 96-well format, to barcode essentially every individual, and then to take all of those samples, pool them, and run them on a single lane of a sequencing machine. And so when we do this, and this is data that's really from Jay Shanduri's group, we can show that we can actually sequence on average about 50 to 60 genes in families. We can run anywhere between 192 families to 384 families on a single lane of sequencing with high specificity, sensitivity, fast, and cheap. It costs us about $12 to do 50 genes in essentially per individual. So that's a lot cheaper than $1,000, right? And so this is essentially the shortcut of the shortcut, try to actually get to this problem of how do we screen thousands and thousands of individuals effectively for these genes. So here are the results. So remember, we did that discovery on 209 families. We selected our 44 favorite genes, and we then went up an order of magnitude. And we actually resequenced the genes. Instead of 209 families we did the discovery, we sequenced them in 2,446. This took about 3 months of time. And when we did this, we identified 27 new mutations. So a new mutation means it's not seen in mom and dad, but it's seen in a child with autism, corresponding to 16 of the 44 genes that we selected. This was very exciting to us. That's why I bolded it. 60% of those new mutations were gene killing. And we would have expected, just based on random, that number to be about 6 to 10%. So a significant enrichment in terms of actually finding gene-killing mutations. And in some cases, like this one, the CHD8 gene that I mentioned before, we actually had 8 new mutations, or de novo mutations. And we had screened already now thousands of controls and had never seen new mutations in this gene in the controls. So this is very much deja vu from the CNV world, but now specific to a specific gene as opposed to a region. So I'm not going to go into detail on this either, other than to mention that this wasn't the only gene we found when we resequenced. So this gene, CHD8, 7 of the 8 of the new mutations were gene-killing mutations, not seen in controls. A gene that's important in terms of synapse function, called GRIN2B, we had 3 out of 4, which were gene-killing mutations, never seen in controls. DYRK1A is a gene that actually is thought to be the gene in one of the 2 genes important for neurocognitive dysfunction in children with Down syndrome. So those kids, as you know, have an entire chromosome duplicated, an extra chromosome. We found 3 out of 3 loss of function mutations in that same gene in kids with autism. Right? So this is from that set of 2,000 that we resequenced. And TBR1, which is a very important marker of early neuronal development in the developing neurons of the brain, we found 3 out of 3. So of the 5 genes now that reach individual significance, they account for, we estimate, about 1% of autism. So some of you will be sitting there going, gosh, that's nothing. I'm telling you, this is how the battle will be won for the genetics of autism. It's going to be proving 1 gene at a time. And think about Chris's case. If we now find patients that actually have CHD8 mutations, loss of function, we can say something to those 8 families, or at least 7 families, these 3 families, these 3 families, and these 3 families, which we couldn't have said before. Coming back to that yarn ball, when Brian did this experiment, he selected his genes that were his candidates, half that they were in the network and half were outside of the network. 16 out of 17 of the gene-killing new mutations were genes that were part of the network. In fact, we only saw 1 gene-killing mutation in a gene outside of the network. So for us, this is important because it says that that protein-protein interaction network that I described is probably very important for the developing autism or preventing autism when the genes are normal. What does that gene do? I used one example, CHD8. The gene is actually on early in development and specifically on in neuronal cells and cells around the neurons. The gene interacts with another gene, or at least at the protein level, called beta-catenin. Beta-catenin is important for turning on genes. And what CHD8 does is it binds to beta-catenin and then binds to another protein called H1, and then it shuts down genes. And you might think, well, why would that be important? Well, for any one of your cells in the brain or your eye to develop, certain genes have to be turned on and certain genes have to be turned off. And so the idea is by mutating CHD8, the correct genes aren't turned off, as they should be in the developing brain. And this is a simple model, and a lot of people are testing this right now to see if this is true, but the data suggests that, in fact, this particular gene is a kind of a global regulator early in development of the human brain. The other cool thing, and I want to mention, is we went back and we looked at the CHD8 patients. And we said, is there anything similar about those patients with respect to their phenotypes? And so what I'm showing you here is head circumference. This is standard deviations. So the mean here for the normal population is the dotted line is actually here at 0. This is just showing you a distribution in terms of head circumference. So it's the distance around the head. And what we noticed with our CHD8 kids, and even from the perspective of autism, is that they had larger heads than the majority of kids that had been seen. In fact, 6 of the 8 kids would be classified as macrocephalic. That means they have a larger head, 2 standard deviations than normal. Here's that gene DYRK1A. And DYRK1A, you can see, is the opposite. See that the kids for DYRK1A, we had 3 kids that had loss of function de novo. They had smaller heads. In fact, this is considered microcephaly or microcephalic. What's interesting, and I think actually is important because it tells us that the gene is probably very relevant. It's been known for quite some time that when you mutate this gene in a fly, it results in a phenotype known as mini brain. So the flies actually have smaller brains as a result of mutation of DYRK1A. And our kids with autism have microcephaly, similar to what you might expect. In mice with mutations of DYRK1A, they also have essentially smaller brain volume. CHD8, as I told you, interacts with this protein called beta-catenin, and it shuts it off. And so what you might expect is, interestingly enough, is that if you actually overexpress beta-catenin, that you might actually change brain volume. This experiment was done 6 years ago by Chris Walsh, and he showed that if you continually express beta-catenin, essentially the brains become essentially bigger in mice. The mice begin to gyrify, so they start to get convolutions in their brain. They don't get smarter, but they actually get convolutions as a result. When we look at, we've identified now 3 patients from our autism collection with mutations, loss of function, new mutations in beta-catenin, and as you might expect, they actually have smaller head sizes. So you might be saying, well, why is he going on and on about essentially head size? Well, if you think about this, this is beginning to actually now phenotypically break down autism from just being generic autism now to being CHD8 patients with larger heads, essentially DYRK1A patients with loss of functions with smaller heads. So it's a very specific type of autism, at least at the phenotypic level. So encouraged by this, we set up this, which, realizing that we now have the technology for actually resequencing thousands of patients, we now had candidate genes for testing. The one thing we didn't have were 10,000s of patients. And we realize now, and this is, I think, a really important message that I hope I convey, is that any individual lab, even some of the biggest labs in the US, do not have enough patients to prove pathogenicity of most of the genes that we're going to discover associated with new mutations. What we need is really 20, 30, 40,000 patients with autism so we can systematically target those individuals and try to identify the 0.1%, the 0.2% with a specific genetic lesion. So over the last year and a half, I've been spending a lot of time essentially trying to network with a lot of investigators in Europe and Australia. I'm hoping this will actually foster some connections here in the Middle East to put together a network. We have about 21 research labs and diagnostic labs around the world that are actually contributing their DNAs with appropriate informed consent for targeted resequencing, discover, and test mutations related to both autism and developmental delay. So each of the groups, I'm just putting up here with numbers and dots, has contributed a minimum of 200 to about 500 individual DNAs from children with autism spectrum disorder or developmental delay. And the most important thing, we've gathered about 10,000 patients now. The most important thing is that if we find something in these patients, they can be called back in, and the parents can be called back in, and we can study the patients that actually have these mutations. It might sound trivial, but essentially one of the biggest problems with most autism research is that in genetics, we can't go back to the patients. So in a clinical setting, you can if you have appropriate consents in place. And so this I've called the ACID network, or Autism ID network, which is trying to connect families really across the world for testing for these genes that we now think are great candidates, but we want to prove that they're actually relevant to autism. And so this is some of the results. So just like the CNV work I showed you from 2006, 2007, we're now doing this for the genes that we've discovered by exome sequencing. So these are patients that have CHD8 killing mutations that are by and large new. We've identified now 3 disruptive gene-killing mutations based on screening 8,813 patients. We found 0 such mutations in 8,792 controls. You can do the statistics, but you probably don't need statistics to tell you that this is really significant. So these are some of the patients that we've seen. 87%, remember we're screening more broadly developmental delay now, 87% have a diagnosis of autism or autism spectrum disorder. Interestingly, 80% actually have macrocephaly. And macrocephaly becomes obvious very early in development. So we now have longitudinal data on several families. And these kids are actually at the 2nd, 3rd standard deviation within the first month of life. So these kids actually have a very large head very, very quickly. 73% have gastrointestinal problems, and we're talking severe. So periods of constipation followed by diarrhea, followed by constipation for long periods of their lives. This is higher and it's significant. Autistic kids tend to have problems with digestion, but this observation is actually significant even based on the baseline of autism. And this is borderline, it's not quite significant yet. 67% of the patients that we've identified actually have severe sleep problems. So they can go, the reports that we're getting from the clinicians is the child can go 3, 4, 5 days without sleeping a wink. And to the point that some parents are actually locking the doors of their houses so that because they can't stay awake any longer with these kids. They're afraid that something bad is going to happen. The last thing that we're doing is we're now mutating other organisms to see if they can actually mimic some of the effects of these. So this is unpublished data from a postdoc of my group. We basically looked at the expression of this gene in a fish. This is a zebrafish. And what's interesting here, the dark shows where the gene is actually expressed. And you can see that the gene is expressed fairly broadly early in development, but as you move to a kind of a later development, it becomes restricted to the head, what we think is a developing nervous system. And if you look carefully, you can see that there's a little trail of signal here which corresponds to the gut of the actual fish. And so my postdoc, when he actually mutates this in a fish, and he actually measures the distance from eye to eye, so this is called the intercusp distance between the eye. This is the mutated fish. This is the control fish. The fish actually have bigger heads, about an increase of about 15%. So we think we're actually now beginning to replicate the expansion of brain volume, potentially, in these fish. And in a set of experiments, which I think are super cool, I'm not going to go in great detail, we've actually now been able to model the gastrointestinal defect as well. So what you're looking at here are fish. This is about 6 days post-conception. And the experiment that's being done is to take control fish and mutant fish. We've actually mutated that gene, CHD8, and then stuff in their mouths fluorescent beads, or fluorescent microspheres, I think is what they're called, and then measure how long it takes for the beads to pass through the digestive canal. And so you can see this little light here. This is actually the beads going out to the fish in zone 1, 2, 3, and 4. And this is the bead leaving. So the fish has defecated this bead. And you actually can see a significant difference in terms of the mutant fish versus essentially the normal fish. More of the mutant fish have their beads stuck in zone 1 and zone 2 compared to the normal fish, which pass these usually within 3 to 6 hours of the system. And in one last experiment this is again unpublished, we asked the question, well, what's going on with the gastrointestinal problems here? This is staining of neurons. So these little lights that you see here are neurons around the gastrointestinal canal or the digestive canal. They're called enteric neurons. And so a really important part that most of us don't appreciate is that we need a pretty robust nervous system to actually pass food when we digest. And what we can see here in control fish versus mutant fish is that the number of enteric neurons looks like it's significantly reduced. So in the brain, it looks like there's perhaps an overexpansion, and in the developing gut, it looks like the neurons are not getting to the right place, or there's too few being produced. This might explain that constipation diarrhea effect that we're seeing in the patients. But I think all of this combines really tells us that we have really a path for actually identifying genes relevant to autism. We're identifying new genes, new families. This is another gene that it's published, I think, 2 days ago, a gene called ADNP. These are new mutations. We now have 10 new mutations in 5,000 patients, 1 in controls. 9 out of 9 are new mutations that are sporadic, and they're all loss of function or disruptive mutations. These are what the kids look like. I actually think they look like angels, to be honest. When I saw these first pictures, they looked like the cherubs that were described years ago. I think they're beautiful children. They have very classic kind of eyes. You can see these are unrelated individuals. You can see something here. They actually have smaller heads. It might not look like this than normal, but they have something very unusual, which is this almost like a bulge, frontal bossing, it's called. So they have a bulge that's protruding over here. And it looks as if we've identified another gene with a very specific set of phenotypes. It's subtle, but it seems to be reproducible in the patients that actually have this particular mutation. Interestingly, we don't have to make the mutant. The mutant was already made years ago in mice. And when you knock out this gene, this is the name of the gene. It's a homeobox-containing activity-dependent neuroprotective peptide. Human geneticists are terrible at coming up with names for genes, but this is the name of this gene. A knockout in mice results in a failure for neural tube closure and death, premature death. But remember, most humans actually just have one copy. If they're autistic, just one copy knocked out, and most humans are totally fine for both copies. And experiments that were just published, a few years back, people actually investigated the heterozygous mice, and they showed they actually had learning difficulties, and they had problems in terms of reduced cognitive function. So I think this is another example, just 2 examples of 500, where we actually are starting to get the evidence that the gene is pathogenic when mutated. And this is the type of data, coming back to Chris's story in the beginning, that we want to have before we tell a family that their child is going to have autism or not. We need to actually have both statistical significance, that requires screening tens of thousands of patients, and proving that the mutations are found enriched in autistic kids, and then seeing the phenotypes, the effects of mutating that gene in model systems. It's a lot of work, but I've been told there's lots of grad students that are interested in this kind of work. So in conclusion, large deletions and duplications are enriched in children with developmental delay and autism, 14% of cases with developmental delay, about 7 to 8% of kids with autism spectrum disorder. Those are just for the very big events. Gene sequencing is fantastic, and even exome and genome at $1,000 is good for identifying candidates. But to screen and sequence 20,000 patients at $1,000 or $2,000 a pop is still a lot of money. So we need methods, and the methods have been developed to actually target the genes that we discover as candidates. New gene-killing mutations are enriched 2 to 3-fold in autistic kids versus their unaffected siblings. 80% of the new mutations are coming from dads. 60% of the deleterious mutations that are being transmitted are coming from moms. So both mom and dad kind of share in this. And I think the really exciting thing to me is that the specific genes now are really identifying specific subtypes. At least we believe we're starting to see specific subtypes, allowing us to really break down the umbrella of heterogeneity. And if you think about this, this is very analogous in my mind to where the cancer field was, for example, 30 years ago, where people would talk about cancer as being one disease. And we now know that cancer is really hundreds, maybe thousands of different types of mutations. And why this is important is that we don't treat all cancers the same way when a person is diagnosed with a specific type of cancer. And I believe this is the same thing that we're going to see with kids with autism. We're not going to treat kids with autism the same way based on the type of mutations. You can imagine if you have a child that has an undergrowth, i.e. a smaller head, or an overgrowth, that the therapies that might work for one kid will not work for the other kid. So I think it's absolutely critical that we really get to the bottom of the genetics. So I'm very much a the glass is half full kind of guy. So take this with a grain of salt, because not all of my colleagues agree that we've made this much progress, but this is where I think we are right now. This is where we were in 2007 in terms of the pie of genetics, so the genetic component of autism. So remember, that's about 60%. We actually only explain maybe 10%, 12% of the genetic basis of autism. I think now we're starting to get around the numbers of like 30, 40%. And really, I think the debate is how much of this de novo disruptive gene-killing mutations can we account for in terms of autism? So that's unknown. But there's obviously an environmental component, and I think that this is like the first step for really sorting out, there should be environment in here, for really sorting out the environmental effect in terms of developing autism. We need to actually understand the genetics of it first to really understand the impact of the environment in terms of this. We have a very simple model. It doesn't get much simpler than this. To develop a human brain, you need many genes. I think everybody would accept that. You need many genes, specific genes turned on at specific times and places. And at specifically early in development, we believe probably around the 11 to 20 week stage, there are certain genes that have to be there in the right amount. And I think those are the genes that we're hitting right now. The DYRK1As, the CHG8s, these are master regulator genes that have to be on at the right time in the right place, or you're going to develop an autistic brain. I think these are important genes because they mean that these genes are probably going to shed light on other genes that are going to be relevant for understanding the pathways leading to autism. The autism genes, we're breaking them into groups. So this is just now a network, again, a little bit more easier to read, showing you all the loss of function mutations. We're starting to group them into pathways, some related to synapse function, so the formation of synaptic structures in the developing brain, genes important in terms of cell proliferation or chromatin regulation. These are the types of genes that we're seeing and their connections. So all the red dots here means that we've seen usually multiple loss of functions in these genes. If we combine all the studies that have been published to date, about 60 to 70% of all loss of function mutations are one step or two steps removed from these central core pathways. And the reason I show you this kind of busy diagram is that while it may be 500 to 1,000 genes, the hope is that it's going to boil down into a half a dozen or a dozen pathways. And it's the pathways that I think are the ultimate target for treatments. Treating the pathways and trying to ameliorate the lost function and what it does in the context of the pathway is the key, I think. And finally, the idea of what we're promising. Well, this is maybe more should be prospects and hopes, maybe as opposed to promises. But this is where we are. We've gone from now mutations to identifying genes. The CNVs were particularly important for telling us that it's not just inherited, but it's new mutations, that it's dosage imbalance. The genes are actually telling us something about the proteins, which are telling us something about the networks. Diagnosis can obviously begin here. Once we've proven CHG8, GRIN2B, DYRK1A, when patients are tested, we can actually identify them earlier as having a high risk of developing autism. So that's an immediate benefit. And this is the hope down here is once you get to the networks, you're going to develop drugs, you're going to develop other small molecules that will actually lead to better therapies. So if these are the genetic types of autism, someone would say, well, why does it matter if I have type 2 versus type 102? I think it's because of this. Two reasons. One is I think the therapies for type 2 and type 102 will be different, as I already mentioned. But something I think that's much more practical is in the diagnostics. Once we start grouping individuals based on their genetic etiology, we by definition connect the families. So we've seen this for CNVs. I can't tell you how many times a parent has come up to me and said, you know what, my child was never like anybody else in those autism support networks or autism groups I went to. My child was always different. And when you actually then tell them that they have a specific mutation and they find other mothers, and this happens all the time, we call them lion mothers because they won't be stopped. They will find the other families that are out there. All they need is a name and they can go after the clinician and get the information. The families start to communicate among themselves. And I think this is very powerful. And I think it's even the thing that the clinicians don't realize is families start sharing information and you have 45-year-old mothers telling a 20-year-old mother how to deal with the sleep problem, how to deal with the constipation issues, long before there's any therapeutics. And I think this is probably one of the most important things we can do is try to connect the families based on really certain genetic causes of autism. So this is the last slide. I just want to kind of put a plea out for this. I think the power of actually solving this puzzle of autism isn't just the advances in genetic technology, which have been absolutely fantastic and critical. They've allowed us to chip away piece by piece, and this globe should probably have hundreds of little puzzle pieces within it. But if there are 500 genetic causes of autism, at a minimum, it is absolutely critical that we network the researchers, the clinicians that are seeing the patients, and finally, and I think most importantly, the families. We've got to be able to network all the way around to have this open line of communication to really understand really the subtypes of autism, but actually ultimately develop the cures or the therapies that will improve the lives of these individuals. So, if you know individuals that have collections with autism DNA samples and they're properly consented, we would be happy to start to sequence them and try to chip away at the 0.1%, 0.2%, 0.3% of genetic causation. So these are all the folks. I'm not going to go into all the names. I want to acknowledge this guy here, Brian O'Rourke. I should have put a star by his name because he just left my lab to start off his own lab in University of Oregon. But these are the people who actually have now started their own labs and that contributed to the work. Also, Jay Shanduri, he developed that new technology for resequencing, just the actual protein coding bits. At Duke, Nico Katsanis actually did some of the zebrafish work that I showed. A lot of great clinical colleagues. I'd actually have to have 2 more slides for all the clinicians. So this is no aspersion on them, but they're all part of the network, and we always include them in our papers. But most importantly, definitely not last on this list, is really the patients and families. Without their contribution, without their involvement in this research, without them allowing us to follow up, without them allowing us to share their photos, we would not be able to do the research that we do. We wouldn't be able to actually kind of bring full circle on this genetic basis of autism. Thank you.
Hi, thank you. One of the first things you mentioned was about the gender bias, and I was wondering if there was any way you could link some of the stuff you talked about later on to that first idea of the 6-to-1 ratio in some cases.
Yeah, so we think it actually relates to the fact that mothers can actually carry more deleterious mutations and not show severe outcome. So it's not as if the mothers don't carry them, they carry them, but they don't manifest severe autism spectrum disorder. And this is the observation, that's what we've observed. So mothers are more likely to carry and transmit.
The explanation of why.
So why? So it could be something like this. This is a hypothesis. Mothers essentially have 2 X chromosomes. There's about 1,500 genes on the X chromosome. About 2 to 5% of those genes are important in terms of developing the brain. Females that actually have 2 X chromosomes are more genetically buffered for those mutations that occur. And so for a long time, people looked for an effect on the X chromosome. It doesn't necessarily have to be there. It just means that females, and my wife would totally agree, are genetically more robust than males. And that's because they can essentially compensate for mutations that may occur in 1 copy on the X chromosome genes. And a male is already a kind of a half-hit wonder because whatever he's got in his X, he's stuck with. And so if there's a gene that produces a protein that's part of this network, which there are, then and you have another hit, you've broken the system, right? And so we think it's an effective threshold. Why the ratios are so different for high functioning versus developmental delay is still a bit of a mystery. I think I think it has to do with really the softness of the phenotype. So an Asperger with an IQ of 130 may require less of a burden to actually manifest. But to get a kid with developmental delay, you need a pretty big insult. So that might push females and males closer to one another for those effects. But that part that's just only a hypothesis at this point. Yeah.
Have you considered going back and sequencing the regulatory elements and microRNAs that are associated with these causative genes to see if there are hits in these elements?
Yes. So we're basically working on a proposal right now. I should point out a lot of the funding that we got was actually from New York, from a guy named Jim Simons. Some of it was from NIH, and Jim Simons actually has a foundation called the Simons Foundation, which has collected those 2,800 families that we started this work on. And he's contemplating right now funding research to do full genome sequencing on the roughly 60% of the families that we haven't found anything based on all of our CNV and exome characterization.
What's your prediction there? Like, you know, I mean...
So my feeling on this is that I think when we sequence the genomes, we're going to actually find the same genes hit over and over again, but it'll be regulatory machinery as opposed to protein coding, because we already know that those genes are super sensitive, right, to dosage imbalance. So I think we're going to circle the wagons more when we start sequencing genomes. But the important point is that we'll then solve another piece of that pie, right? We'll start to get more information on those families. But that's the logic. I mean, I can talk with you afterwards. We've actually spent a lot of time with the experts and said, can you tell us exactly where the regulatory regions are so we can just target those? And for every investigator I've asked, I got a slightly different answer, right? So I think the right answer is just sequence it all at this point.
Firstly, thank you for your brilliant presentation.
Thank you.
But according to what you said, our focus should be directed on genes only. For example, if there are other factors in pregnancy, during pregnancy, or something like that.
Yep.
The secret of autism lies only in genetics and genes, or can we focus on other factors that can lead us to the reasons?
Right. So the question is it just genes, right? So is there more to the story? And the answer is absolutely yes. There's good data that suggests that certain environmental insults, certain infections, certain exposures with alcohol, certain aspects will actually contribute to a significant risk in autism increase. So it's not just genes. And that's the very opening slide is we estimate more than 50%. And that's pretty much a cop-out saying it's 50% genes and 50% something else for some of these other families. But my feeling on this is because we've come so far in terms of identifying or how to get to the genes, that we should start with the genes first, work out as many of those cases as we can, and then spend more time on the ones that remain for environmental or maybe combination of environment plus genes, right, in those cases. And I think there's going to be a whole category that are more difficult to solve, and those are ones where it's going to be genes plus environment plus maybe some other epigenetic phenomena that goes on.
With all these genes and all the differences that exist, I actually think it's totally right to think of the pathways that are going to be coalescing out of the genes. But there seemed to be, from your story, and this is the naive part, if I got it right, that there is some signal in the context of it's mostly what you presented, loss of function mutations, and also mostly what you presented were de novo, which I think means heterozygous individuals.
Correct.
Right?
Yeah.
So is there something really interesting about the biology of a potential moment in development that is incredibly sensitive? And why are these genes not being buffered, as you said? And is it possible that they're being stressed at the same time? The system is in some kind of a moment of a hit, a double hit, a triple hit, and a stress that leads to the disease. So,
yeah, it's a great question. And so, in fact, there's some papers that came out, not from our group, from other group, that tried to look at this question more specifically by looking at the expression of these genes. So, co-expression networks as opposed to protein interaction. And their data suggests that there is a network, and there's multiple networks, but it's particularly early in development, as I mentioned, this 15 to 20 week window. And it's also in the layers of the cortex when neurons are migrating from essentially the cortical, subcortical layers, ventricular zone to the cortical plates. And if you think about it, this is a very critical time for humans. Because if you look at humans compared to all the other great apes that are out there, the one thing that distinguishes us is our expansion of our neocortex. And that the layers that people have seen these genes expressed in, and the timing, is the time where we have to really move. We actually have to build a lot of neurons and move them to the right locations in the developing cortex. And so, if you think about our brain expansion, and if you believe the fossil record, it's a pretty recent deal, right? So the human expansion is only about 1.5 million, 2 million years old. And I don't think we've reached equilibrium on that deal. And the reason I bring up the kind of the smaller brains and the bigger brains, or at least the micro-macrocephaly, is because I think we are very sensitive because we haven't actually reached a stable point in the developing of the human brain. And so in some respects, autism and developmental delay for humans, I think, is a consequence of our adaptation to essentially enhanced cognitive function. And so we pay the price for that by actually having a sensitive system and children that have disease.